Dr. Barbara Methé is a microbiologist and a bioinformatics expert who's really contributed significantly to microbial genomics. So how do we study the microbiota in terms of their genomic sequences? And the key contribution have really been in identifying how these microbes are engaged in things like bioenergy, bioelectricity, bioremediation, and really trying to these seminal discoveries have really pointed to how we can use a genomics-minded approach to understand these complex host microbe interactions. So with that, welcome.
Awesome.
Thank you.
All right. Good stuff
So
here.
take them just a little bit to get the slides.
Yeah, no worries. Okay, so the challenge that I received for this talk was to kind of summarize what are some of the important concepts, challenges in the microbiome space. And so I will attempt to do a little bit of that for you today, and I will nominally wrap it around a little bit the concept of precision medicine, which we've heard a little bit about already. So let's see how we do here. So of course, the concept of precision medicine, as we have heard, is you want to be able to take information about an individual, so hopefully deep phenotyping. This would include information about a person's genetics, about their lifestyle, about their environmental exposures, as well as information about their immune system and so forth, and include the microbiome. Crunch all of that data, and hopefully we get to a place where we're able to define underlying molecular mechanisms of health and disease, that we can, for that person, understand better what are their health risks, as well as design targeted therapeutic approaches, and are there preventative mechanisms and preventive strategies that we can tailor for that individual. And so in terms of the microbiome, we specifically want to understand things about not only who is there, but functionally, what is the microbiome doing in terms of metabolite production and so forth. How can we use that in precision medicine approaches? So with that idea, we've heard a little bit about this in the first talk, and so currently we have a lot of promise and possibilities, and we've heard about those things. We're not really at a place, quite frankly, where we can really use the microbiome in a precision medicine scenario. Again, we have prebiotics, probiotics, et cetera. They're available to people, but the marketing, quite frankly, is in front of the science. And we do not have good, solid evidentiary science for a lot of these therapies. So why is that, and maybe what are some of the things we can do to deal with that challenge? So I put this slide up here just as a reminder that the microbiome, it exists, we think of it a lot in the human space. That's the part that a lot of us study, and that's what we think about. But I put this up here to remind you that the microbiome is everywhere, right? It's part of our built environment and our natural environment, and microbes really are the fundamental drivers, for example, of all the biogeochemical cycles on our planet. So when we think about the associations of the microbiome within, we also have to remember that there's a connection to the outside space, and microbes exchange with our external environment and are influenced by that as well. So when we think about, for instance, precision medicine type strategies, understanding what people's lifestyles and exposures are is going to be a very important piece of that puzzle. So One of the challenges then in terms of studying the microbiome is really we need to look at it in the context of microbial ecology. Microbes form communities, right? They don't live in pure cultures. They form different assemblages. These organisms interact with one another. They sense and respond to their environment, whether that is in soil or water, or whether that's inside of our human body. And so as a consequence, we really need to be able to take the multi-omics approaches that we now have available to us and apply them to the microbiome so that we can study it as a community as a whole. And then the challenge, of course, is to be able to relate in the human space, to be able to relate that information to information about the host. So some of the important emergent properties that we want to look at are not just taxonomy, but we also want to understand functionally what is the microbiome doing. And so we need to also think about how microbes interact with one another. There are competition, there are cooperation strategies, all of which are very important. And of course, if you are a microorganism, fundamentally, you have to be able to find a carbon source, and you need to be able to conserve energy to support growth. So understanding the flow of carbon, nutrients, and energy through a microbial system is incredibly important if we want to be able to manipulate them in ways that are beneficial to us. So again, whether that's something that's happening in the environment or whether that's something that's happening in a precision medicine situation in our own bodies. So in my world, we work a lot in clinical settings where we use a lot of large observational cohorts for a lot of the work that we do. And these are very important because ultimately, in order to place an individual in a precise way, if we care about precision medicine, we have to understand populations of people in order to know where does someone fit in the range of health and disease. And so here I've just given some of the basic steps that we tend to go through and use when we're doing this type of work. And so obviously it starts with your study design, and you can never put enough effort into that. We also have to be able to collect samples that are reflective of the microbiome. And then we can use different multi-omics approaches, so the idea of systems biology that allows us to study the microbial community as a whole. And when you get that type of data, it tends to be rather noisy, right? And a lot of times the signals you're looking for in that data can be quite small relative to the background noise. So there's a lot of quality control and processing that needs to happen. And then there's ultimately data analysis and integration and interpretation. So, and we want to avoid the GIGO idea, the garbage in, garbage out, right? If we're really going to be able to use these large cohorts, well, we have to really think carefully about all of these steps. So one of the projects that I'm working on right now is this project called nominally MedBio. This is here where we're crowdsourcing across a multitude of physician-led cohorts here in the UPMC system. And our goal here is to collect information not only about the human, particularly being able to sequence human genomes, but we're also taking microbiome samples, and we're using those samples in various ways to study the microbiome. And we also have access to the electronic health records and other information about these individuals. And so the goal here is to be able to take all of that information and ultimately integrate that and use that in a number of different ways. This becomes now a framework where not only can we look at host microbiome interactions, but it also becomes a platform now to be able to layer other types of data onto this framework. And so currently, for instance, we're working on integrating information about things like socioeconomic factors, as well as air pollution data and other types of environmental exposures as a part of building this framework. So, again, another part of the of this challenge is ultimately, right, we have the high throughput omics techniques that we're using for many of these studies, but I always point out that ultimately what you ideally want to do is to be able to have experimental validations of hypotheses that you've gained from your high throughput omics studies. And you want to be able to test those, and ideally that's the kind of information that you want to be able to put back into a lot of your to your high throughput techniques and through your data mining and data analysis. And so I've pointed out some different platforms that we are working with in various ways, and they include things like 3D organoid systems. There are also synthetic biology approaches that are becoming a really nice complement to a lot of work in the microbiome space. These become ways that you can actually test principles about how microorganisms interact with one another. You can change the genetics of the organism so that you can try to engineer it effectively to do something you want it to do. You want it to make a particular metabolite. You can put it under control such that you can switch that metabolism on or off. So it's effectively like building a circuit. And you can also begin to learn a lot of principles about how microbes work through those kinds of strategies. And then there are other platforms such as spatial genomics to be able to interrogate things like how do microbes actually interact with one another? What do they look like in a particular space, and be able to see those relationships more clearly. And then, of course, we've heard about gnotobiotic mouse models. So these are all important for this type of work, and again, you want to be able to integrate that into the high throughput genomics. So one of the places where certainly in a clinical setting where we need improvement and are working on improvement is this idea of doing different real-time sampling and monitoring. And there are lots of different developments that are happening in this space. In our case, we're working on a ventilator system that allows us to collect exhaled aerosols from the lung. So we work a lot in the lung, and being able to get good microbiome samples from the lung is a huge challenge. And so we're working on this idea of being able to collect exhaled aerosols. In this case, we're using it to look at patients who are on ventilators, for instance. And so this is a way to be able to real-time monitor and assess the microbiome. We're applying that in some different settings, including in the intensive care unit. And what we're doing is being able to take these samples, and we're taking advantage of improvements in different high throughput technologies. So here we're using this third generation sequencing platform from Oxford Nanopore, the MinION, and we can be able to get real-time information, metagenomics information about these patients, and we're trying to use that information to in particular to assess what pathogens they may be colonized with, what antibiotic resistance genes may be available, and can we improve treatment for these individuals in that way. So we get lots and lots of data, and we need ways to be able to analyze this data. We get large volumes of data, and in particular, again, it's very noisy. So this is where the machine learning, the artificial intelligence and machine learning opportunities become very important. And so I just put this slide up here. This is not a comprehensive view of all machine learning, but just to point out that typically it's divided into two kind of topics right now, that approaches that people use for studying in the microbiome space. We have the concept of the unsupervised learning and the supervised learning. So the unsupervised learning allows us to do exploratory work, and the supervised learning is the place you go when you want to kind of get more predictive models. And kind of coming into the space now are different deep learning options. And so, for example, this is an example of an artificial neural network. So this is becoming a new space where we can gain more information potentially in terms of being able to integrate our data and to be able to make more predictive models. However, these types of approaches, they require very large amounts of data, and that's still a problem in the microbiome space. You know, these are very kind of greedy. These are greedy techniques. You need a lot of data, and that's still a big challenge in the microbiome space. You also, in terms of computational hardware, you really have to be in the graphical processing unit, GPU space, as opposed to the typical CPU space. So that's another sort of challenge. And then I'll just note here that in order to do these types of AI approaches, there is a machine learning life cycle that's really important here. I won't spend a lot of time going through all of the parts here, but it's very important not just to think about training your model and validating your model, but also to think a lot about what is the data you're putting into that model, what is the pre-processing, the feature engineering and selection that you do before you even run the models. It's a very iterative process. And you're always making trade-offs between things like bias and variance. And so you have to be very mindful, because these can be very black box initiatives. And so you have to be very, very careful and mindful of following good principles in order to produce the best models possible. And so then I will just wrap up here with another area that we're working on now, and again, getting some of the multi-omics data that we've been generating. This has been very helpful for some of these constraints-based metabolic models. So these are mathematical approaches that now we're going to model metabolism, right? And so we can use genome reconstructions to do this. And what's really important about this is you can run lots of simulations about different scenarios if you restrict certain carbon or nutrient sources, and you can ask questions about how does the microbiome respond. And so this can be a very informative framework and also help guide a lot of experimental work as well. And so I think I've talked about some of these challenges. Again, I just point out that we need more N, we need more information from more individuals in order, again, to be able to get to the precision part of the medicine that we're looking for. And there's a number of challenges with the models, but again, those are things that are certainly coming. But you need to be mindful of them, because these, in particular, a lot of these AI models, they do tend to be very black box. They don't always reveal sort of how they're giving you some of the information that you're receiving. And also, the compute architecture that we use is really going to also be, undergoing a lot of changes as we go forward, and going to be very important to supporting the ability to use the volumes of data that can now be generated in useful ways. And so I'm going to stop there, because I think I'm probably over my time, and I'll just thank you for your time and attention. Thanks.